my project this summer was light sheet fluorescence microscopy and patterns of structures in the kidney. And I did this summer research in the Jane Lab at Washington University Med Campus. So my overall question for my project was, what patterns are present in healthy versus diseased kidney tissue structure? And so the objective for this summer was to specifically identify patterns of structures in a reference tissue for more of a healthy reference that could be compared to other biological factors later on. And so how these patterns were identified was through light sheet fluorescence microscopy and staining these kidney tissue samples with different markers to make certain cell types visible. So just a little overview of the kidney for those who are not familiar. It's responsible for filtering the blood in our body. And so it filters this blood and then reabsorbs necessary substances back into the blood while secreting and filtering waste out through urine to be excreted from the body. And this is a little graphic. This kind of zoomed in image of the nephron, which is a functional unit of the kidney, gives a little bit of a preview of what kind of structures that we can stain for and later image using light sheet. So this is a little background of the light sheet fluorescent microscopy workflow. First, but after obtaining your tissue, you have to fix it, and cutting little samples that have to be cleared. So what clearing is essentially making the tissue sample as transparent as possible so that when it is time to stain it for the cell types and structures that you're looking for during imaging, that it's as transparent as possible and makes that process easier. And then next to stain, we had antibody labeling, which involved attaching a fluorophore to secondary and beforehand a primary antibody. And then we have imaging with our light sheet microscope, which later goes through image analysis and quantification of these structures that we're seeing. And then these are little examples of our tissue samples that we would get and would later go through this pipeline of light sheet microscopy. And so we can see some healthy samples right here. And then there's a tumor. This was an image, but this is just a reference of what these kidneys look like, or these samples look like when they're cut out of kidneys that we get in OR. So before clearing the tissue, the sample can look like this. This is just a reference of actually what we were looking at in lab before our tissues went through their clearing protocols. And then the clearing that we did in this lab was actually done in two different ways. So we had active clearing, which was more of an active process, but it was not something that I had hands-on involvement with. So these samples were sent to the WashU Imaging Center, and the timeline for this process was faster. So it took about 7 to 10 days. And then the cubic protocol, which was done here in the lab, I had hands-on involvement with, and it was more of a passive process that actually took longer. So it took approximately 17 days. And here we have images of what samples look like after they've been cleared with the two different methods. So once they were cleared, they had to go through fluorescent labeling, and both of these different tissues that were cleared in different ways went through the same light sheet staining process. To familiarize with the immunohistochemistry that was involved, we conducted some confocal fluorescent microscopy, which was done here in the lab with 2D sections of tissue versus an actual section that hadn't been cut. So we cut these tissues, and these are some examples of the antibodies that we can use to fluorescently label this kidney tissue. And so over here, we have a list of primary antibodies that we can use, and then the confocal tests that I was involved in the lab have checks next to them. And so luckily, I was able to see a lot of different antibodies imaged on 2D sections before I was even looking to stain light sheet tissue. And then all of these antibodies have been used at one point to stain the tissues we were using for light sheet. And one thing that I want to highlight is this NPHS1, which became pretty important to staining a structure in the kidney called the glomeruli or the glomerulus. These are just some examples of the confocal imaging that I was doing. And we have these different colors for each of the structures, signifying a different structure labeled with the certain fluorophore. And we have the image next to the control right here. And next we have imaging that was actually done with the light sheet microscope. So right here we have a snapshot from an imaging software that was used in a lab called Imaris. So we can see the various structures. This sample specifically was labeled with 4 different antibodies, and we're able to see the different colors. And then in pink right here, we have the glomerulus, like I was referring to when pointing out the NPHS1. And this is just a picture of the type of microscope that we use in the imaging center for light sheet. So next we go into more specific quantifications, specific quantifying the glomeruli. So like I was talking about, here's the reference picture again. The glomeruli is where the blood goes to first in the nephron, and it's where all the blood is sent and initially filtered. So this is what that image looks like from the slide before, when you mask just the glomeruli and pretty much block out all of the other structures that were stained for, and just focus on this one here. And so now we have some quantification values for the glomeruli. We were able to use our imaging software to obtain volumes, surface area, and sphericity of the glomeruli. And just give me a second here. Oh. Okay, so this was done using a segmentation algorithm called CellPose. So after these glomeruli were masked, they went through an algorithm, and then they ended up looking something like this before we could obtain these values. So going back over here, we had volume, surface area, and then sphericity of the glomeruli. Because they're ultimately round, we had these values for how spherical they were on a scale of 0 to 1. And if you notice this outlier right here, this is actually interesting. So this smallest volume and this least spherical glomeruli is actually the same one, which is pretty interesting. And so we had these samples from about 30 glomeruli from 3 different viewpoints of the same kidney sample. So for the conclusions of this project, in the lab, we were able to establish an effective pipeline between the initial tissue acquisition and preparing the tissue for light sheet to actually using light sheet for data production and structure analysis of quantification for these values of volume, surface area, and sphericity. And then we conducted light sheet microscopy for 4 different markers at a time in these samples at over 4,000 micrometers deep, which is actually pretty impressive considering that when a confocal microscope is used, you can only get up to 1,000 micrometers deep, even considering that, which is affected by different variables that can make it less. So 4,000 is quite a feat. And then we created metrics for comparison to a healthy glomeruli and what that would look like compared to a glomeruli later on that could be quantified from a diseased kidney. Some of the limitations of this project was the ability to more easily mask a circular image versus any other type of structure. So because glomeruli were more spherical, it was easier to isolate them. And when trying to quantify different structures in the future, this might be a challenge. And then glomeruli at the edge of the images were cut off, so we couldn't really use those values because getting a volume of a little corner piece that was cut off wasn't really productive. So for future directions of this project, for the next 2 weeks that I'm still here, I'm going to extend this quantification to even more samples to get even more data for glomeruli. And then for this pipeline in general, it could be used to evaluate different samples of different biological different biological variables like sex and disease, like we were talking about before, and comparing them to different reference kidneys. And then in general, this technology could be used to quantify different structures in the kidney, but also distances between different structures, such as the distance between a glomeruli and a neuron in the kidney. So that was all that I had today, but I wanted to thank the people in the Jane Lab for everything that I was able to do this summer. And if there are any questions, I will welcome those.